All right, hi everyone. Yeah, welcome. So I have the honor of introducing to you Thierry Emonet, who's visiting us from Yale University. He's a professor there in Department of Molecular, Cellular, and Developmental Biology, but I think for the sake of this audience, he's also a professor there in the Department of Physics. Thierry, his interest, let's say, shifted. He started out as doing theoretical astrophysics for a number of years. I believe went on did postdocs in that subject, and somewhere along the line started getting excited about questions in biology, in particular questions about computation, how living things can actually compute. And so his lab now is looking at exploring this question over a range of different systems, looking at how bacteria and populations of bacteria interact and behave, looking at how more complicated eukaryotic cells come together to form larger organisms, and understanding things like how senses and things like odor and olfaction are actually acted on by model systems. And so he's going to be talking on that later topic here today, but there's a wide range of, I'd say, very interesting quantitative biological questions that his lab studies that you could also pick his mind to afterwards during the coffee as well. So anyways, so I'd like to thank him for visiting, and I'll pass the mic.
Okay, thank you so much for. So thanks for having me. Having an awesome time today. I met with so many interesting people, and I think I was invited to talk mostly about probably information processing in bacteria and stuff like that, which is closer to physics. But I got really excited to talk about this because we just made a discovery, which I think is important and cool and has several implications. So I'm excited to talk about that. I hope it's okay. But so as he was saying before, my lab is interested in biological computation. So what type of computation do biological systems perform? And how do they implement this computation using molecules or cells or circuits of cells, circuits of neurons, etc. And how then this shapes their performance at carrying tasks. And as a framework to ask this type of question, we study chemical sensing and navigation. And this is a great framework to ask this type of question because in order to navigate chemical fields, you need to be able to sense, process that information, make some sort of decision, integrate with previous information, then take some action. And whenever you act during navigation, you're going to change the signal you're going to experience next. And so that gives rise to a very rich type of dynamical structure. And as model system, we use two types of organisms in our lab. One is E. coli. So these are tiny bacteria about 2 microns long. They live in your gut. They're in the lake, out there in front. They're basically everywhere. And the study of how those bacteria are able to navigate gradient has become sort of like an hydrogen atom type of problem in biology. It's probably one of the best characterized model system in all of biology, which allows us to ask very precise quantitative questions. And so those bacteria, they move around by rotating flagella, and they perform a random walk because they're too tiny to detect gradient without moving. So on their scale, diffusion will equilibrate any kind of difference in concentration in 10 milliseconds. So they have to move to detect gradient. And as they move in a random direction, they control the probability to change direction. And if life is getting better, they lower this probability to change direction in order to keep on going in the same direction. And you can think about this strategy as a very general strategy, because even us humans, when we get lost, we tend to initiate a random walk until we start to recognize things. And then we recognize we keep on going in the same direction. And if you look at flies, larvae, etc., many animals perform this type of strategy. And so in here, one of the latest questions, I'm just going to give you just two snippets of papers that we just published on this. So one, we ask, how efficiently does E. coli use information? There's been a lot of study examining how much information is an organ is able to extract. But here we said, given a certain information rate, how efficient are the organisms at using it? And in the case of bacterial chemotaxis, we were able to measure this very precisely. So we were able to measure the rate of information in bits per second that those individual bacteria are able to acquire as they're navigating. And at the same time, also measuring the speed at which they're able to climb the gradient. And so what's interesting is that you can extract a theoretical bound of the maximum speed that can be achieved given a fixed information rate. And then the question was, given this information rate, how close are you from this bound? And this bound, by the way, has parameter that has to do with the way those bacteria navigate, and we can measure those parameters. So this is actually an experimental measurement. And then these are also experimental measurement. And what we found was that two things. First of all, those bacteria are able to navigate using 1% of a bit per second on average. So on the scale of 1% of a bit, not 1 bit per second, but 1% of a bit, which is really low. And they're able to perform at about 65% of the maximum that would be possible. So that means that they're actually very efficient at using this tiny amount of information they're able to extract. It also shows that the amount of the efficiency and the amount of information they're able to extract will put a bound on the possible performance that biological system can carry out. Another type of question we ask with this is, what happens when those bacteria are navigating as groups? So if you look at a population of isogenic population that all have exactly the same genes, when you look at the behavior, they all look slightly different. And that's because gene expression is noisy. And so each individual cells will have a different number of the same protein. So the protein that they use to detect the signal, one bacteria will have 6,000 copy, another one 5,000 copy, and so on. So you have a distribution. And so this distribution in the parameters of these active particles, if you want, will map onto distribution of performances when they're climbing a gradient. And actually, we showed in previous paper that the individual bacteria, some will navigate 10 times faster than others. But at the same time, they can do this kind of behavior. Here, all of those bacteria, they're moving together because they're consuming some attractant in the environment, which makes a front. And that front is moving away from them because they're consuming it. And they navigate that gradient they are generating together. No? The question is, how can you have all of those cells navigating together at constant speed when in your group you have so much diversity in the performance? And what we found is that what happens is that within this group, cells self-organize by performances in a way that's stable. So the ones that are very good at navigating, they end up towards the front, but at the front, the gradient becomes very shallow, so the signal is weak. And the ones that are weaker fall towards the back, but the gradient gets steeper at the back because of the consumption of all the cells in the front. And so it's easier to navigate. And so each cell ends up compensating their differences in performance with difference in position that have difference in quantity of information to navigate. And that leads to this spatial organization, which then is very interesting because that means that even though you have one genome, you have diversity of the population, and that population self-organize as it's navigating through an environment. And in the second paper, we explore what happens when now this group encounters multiple environments. And what we discover is that in each environment, the organization of who is the leader and the follower will change. But how long it takes for this organization, this group to reorganize itself depends on the inheritance of phenotypes from mother to daughter cell. So whenever a mother cell divides, if the daughter cell inherits perfectly the phenotype of the mother, what would happen is that eventually you're only going to have the red cells in this group, because the one at the back tend to fall off slowly, and you're going to purify and remove all diversity. But if you do that, when you hit a new environment, you're not going to have the phenotype that would be best in that new environment. So there's a trade-off between, if you want to navigate quickly as a group through a uniform environment, if you remove diversity, you can go faster. But when you hit new environment, it's going to take longer to reorganize yourself. And the knob, the control parameter that controls this reorganization time, is the inheritance, the rate of inheritance from mother to daughter cells. And what's cool is that all of this is non-genetic. There's no mutation whatsoever in this problem. There's no control, there's no regulation. It just happened because when you do things collectively, there's a selection for the performance of the individual to be able to keep up with the group, okay? And at the same time, there's this generation of phenotype within the group. And that can shape the distribution of individual within the group, just dynamically. Okay, so that's all what I wanted to say about E. coli. I'm happy to talk to you more about it later. I got really excited to talk about this. So these are flies, now they're fruit flies, and they are constrained to walk between two pieces of glass. And then from the left here, we're flowing a laminar wind, but from the top and the bottom here, we have tiny little jets that stochastically turn on and off that perturb the flow. And the dark thing that you're seeing here is basically smoke that we've put to visualize the flow. But then we were lucky, we realized that the fly loves smoke. And so they track this. And you'll see why it's important. So this is one of the first experiments where you can actually see the signal, the olfactory signal that the flies are following and the behavior of the flies, okay? And what I'd like to discuss today is what kind of information do the insects use to navigate odor plume, which has been a long-standing question that started more than 100 years ago. And this is work done by Niraj Kadakia, a postdoc in my lab, and Mahmoud Demir, another postdoc, and a collaboration with Damon Clark, who is a colleague of mine, who is expert in vision and motion in vision, and you'll see why it's important. And that was just published last week. So, okay, so navigating chemical is really important. The odor, smelling, odor, sensing chemical is probably the most important sense in the animal world. And it's also the most ancient. And it's the one that animals use to find food, place to lay the eggs, find sexual partner, etc. Also for destructive behavior, like whenever a mosquito is finding you, is because first it was able to smell you. And it can follow the smell hundreds of meters away from you, will find you. Okay. So now, navigating odor, it's hard because odor are transported by the air, and air tends to be turbulent. And so if you think a fly is here, what the fly is experiencing is actually very sharp, short, random whiffs of odor. And if you study the theory of turbulence, for example, you can actually calculate the duration and intensity of these, and you find that they follow power laws with a very clearly defined scaling law, etc. So the signals that the flies have to deal with are power law distributed, which means there's no real scale to the signal. They can scale over many, many orders of magnitude. And they're interspersed, so do you have periods of blank, and then one whiff, and then nothing, and a whiff again. So how do you navigate that? There's no clear gradient to navigate. No? And so one question that has been there for a very long time is what strategy do insects use to navigate odor plumes? And this has been very important for a very long time because mosquitoes, again, that transmit malaria, for example, that's what they use to find their target. No? So one key difficulty in studying that question is that you cannot see the odor. It's very difficult, and you can try to use odor that are fluorescent, but you have to get to concentration that are so high that basically they become toxic. But there are some experiments that use planar laser illumination, and you can get in physics lab actually a measurement of the odor field, but not simultaneously with the animals. But so in the 70s, various groups had this idea that, so to overcome this limitation, what they did is that they released some pheromone together with soap bubbles, and then let several meters down release some moth and see if the moth could reach the target. And what they realized is that the moth would move straight upwind whenever it was in the field of bubble, which is where the thick line is. And then whenever it would lose the field of bubble, it would go crosswind. And so that suggested that basically a basic strategy to navigate this odor plume is whenever I smell something, I'm going to go upwind. When I lose the smell, I'm going to go crosswind to try to find the plume again. No? Makes sense. And that's the general strategy. It has been recapitulated later in the lab. For example, this is work at Caltech by Dickinson and Van Brugel. They had like a straight plume. So because they cannot see the odor, they have to make a straight, stable plume. And then they map in space where that plume is. And then they manage to track the flight of the flies in 3D. And so that they get these trajectories. And they find the same things, that the flies reflectively turn upwind whenever they encounter the odor. And then other labs also doing same. This is in New York, Kathy Nagel's lab, the same thing. Whenever there's odor, you see that the fly is moving straight. And then when it loses the odor, it can initiate some sort of search. And so in our own work, using this smoke assay, we could, in addition, get this type of data where we see the odor and the fly at the same time. And so we measuring the concentration of odor into this, if you want, virtual antenna right in front of the fly. And this is the signal that the fly is getting. And here you have the orientation and speed of the fly as it's trying to navigate. Huh? So from this type of data, we discovered that the fly were performing saccadic turns. So at random time, they would turn plus or minus 30 degrees. But they biased the direction of the turn upwind, conditional of the frequency of odor encounters over the past second or so. Okay? So they're responding to the frequency of whiffs. But still, in this and from the 30s to today, all of the paper basically point to the same kind of strategy. The only directional cue that the flies are using in this kind of a task is the wind direction. The wind direction provides the directional cue. And the encounter with the odor is a timing information. When should I change behavior, go upwind or go crosswind? Okay? And so we wonder, is wind the only local directional cue available, or is there other directional cue that they might be using? And so the flies, this is the head of a fly. Here you have the eyes. Those are the two antennae. These antennae are covered with little hair. And each one of those hair, there's a sensilla of olfactory receptor neuron that bind odor and generate spikes that are sent to the brain. And there's a distance between those two. And so, of course, people have looked at, well, can I just measure gradient with those? Can the fly measure gradient with those two antennae? And they can. And they actually respond to the difference between the two and turn, in some experiment, more to the left or to the right, depending if there's more odor on one side or the other one. And this was shown by several groups, but in particular, this one is for a case where the flies are flying. So those experiments were done with a straight plume of odor. But when you have this kind of complicated signal, you see that the direction of the gradient is pointing all directions. No? So it's not very useful. It's not a very good indicator of where the source is. So we were wondering if there's not an other type of signal. And you see that, well, we don't really see very well here, but basically, if you were to look at the direction of motion of those packets of odor, you see some sort of regular motion away from the middle line of the plume here. So we were wondering maybe the fly can detect that and use it. So this is going to be the talk. I'm going to talk about, can the fly detect the motion of those odor packets and how they do it and if it's useful or not. And so the first question is, can they detect that independently from the wind? And so first we looked back at our experiment where we have this smoke and the behavior of the fly. And in this experiment, we have a trajectory of a fly. And so along this trajectory, you can measure the gradient in this direction, perpendicular to the fly, no? And so this is the odor concentration. The odor gradient is kind of fluctuating. And also you can measure the odor velocity. The odor velocity here is basically you look at subsequent frame and look at the correlation of the odor signal across frames. And from there you extract a velocity signal, no? And so that's the one that you have here. And then you have the fly orientation and the walking speed. So now with this type of data, you can ask, for example, if the fly was oriented in this direction at 45 degrees up here, and the odor, does it respond to odor concentration? And you see that the velocity, the angular velocity of turning this way is increasing with odor concentration. So if the fly is going this way, and the signal is strong, it turns upwind. So that's to be expected. The same thing for the flies that are oriented in this way, they would turn the other way. But if they're oriented straight, on average, across the whole space, there's no response to the odor concentration. For the odor gradient, the same thing. If they're oriented already in this direction, they don't respond to the odor gradient. But surprisingly, if they're already oriented this way, you see that they respond very strongly to the odor velocity. So if the velocity is in this direction, they turn this way. And if the velocity is in this direction, they turn that way. And so this is just correlation, but that encourages us to try to continue looking at this. So the next thing that you have to do, what we could do in a fly, which is really fantastic, is that you can basically express in those antennae channel rhodopsins. So this is an ion channel that will respond to light. And then when light hits that channel, it opens and makes the neuron to fire. So you can replace real odor with light. And if you do that, you can separate the wind from the odor signal. And you can do this type of thing. So you can now project the plume with a projector and have the flies responding to this type of signals, okay? So now using this approach, we ask, okay, let's remove the wind. Now we have no wind, and we're gonna put bars of light. And those flies, by the way, are blind, so they cannot respond to the light with the visual system. They can only respond to the light with their olfactory system. And so if they respond to the motion direction, when the bars are moving from left to right, they should turn into the bars and vice versa. And that's what we saw. So when the bar were moving this way, the fly oriented in this direction. When the bar were coming from the right, the fly turned towards the right. Like they're moving against the motion of the odor. No? So that suggests that they're actually able to detect the motion, the direction of motion. And you can do control. So when there is no motion, the distribution orientation are basically neutral. You can remove one of the antenna, and then this disappear. So that means that they actually really need to use the two antenna to do this. And finally, you can, so in this, for the neuroscientists here, you have many different neurons that sense odor, and each neuron has a different type of receptor and respond to different types of odors. No? So you can express these channel rhodopsin in all of those neurons, or only a one type. That would be for a single odor, and it still works when you have it only in one type of those neurons. Okay, but still, those bars like this is not a real good proof of are they responding just to the motion or the gradient. So we can do another experiment where we make those bars a bit wider. And now there's a clear prediction. if you so the bars are going to be moving from left to right. And when the on edge, the initial edge of the bar comes, if the fly is oriented this way, if it responds to the gradient, it should turn left, and if it responds to the direction of motion, it's also to turn left. But when the off edge of the bar is passing over the fly, if it responds to the gradient, it should turn right, whereas if it responds to the motion, it should turn left. So there's a clear distinction here. And if you now do that, if you project those bar and you have those fly oriented however, you can plot your data as a function of the orientation of the fly. So if the fly is already oriented perpendicular to the bar, it should be no response because the two antennae will get the same signal. But if it's oriented this way, like here, you should see an opposite response if you're gradient sensing, but the same response if you're motion sensing. And the data is very clear in this case. They really respond to the motion, both at the on edge and the off edge. Okay? So now the next question was, what is the computational algorithm that the fly are using to detect this motion? And that is where that's a place where things are really fun, because there's been a lot of study in neuroscience of how your visual system is able to detect motion. And so a classic experiment, for example, is to put this type of signal. So in this type of signal, you have these random bars coming in, but there's a correlation of the bars from left to right. And in here, if you look at this in the center, you should have the impression that it's moving towards the right, depending on how the projector is working. So is it moving to the right? Do you have the impression it's moving to the right, more or less? I'll do another test with you later for another one. Okay, so this signal is generated like this. You put the bar, and then the next time step, the pixel on the right has a higher probability to be the same color as the one on the left, but not 100%. But you see that there's a correlation from left to right, no? And so if you do an experiment with this signal that don't have net motion, they just have correlation, and the wind, you see a clear response. But now you can do another type of experiment where you put the apparent motion due to the correlation opposite to the wind. And what you find is that this is really messing up with the response of the fly. So they're really responding to these correlations. So now there's another thing you can do, which in the visual field is a classic experiment, which is this one. So which one of those two squares is moving to the right? The top one or the bottom one? All right. Who says that the bottom one is moving to the right? Who says that the top one is moving to the right? Okay, clearly, no? You can clearly distinguish it. The only difference, so here, what we're doing is that in both cases, the correlation is from left to right, but we're reversing the sign. So the phase is inverted. And so whenever you have a black pixel, the next time step on the right, you're going to have a yellow one and vice versa. Okay? And that reverses your sensor's motion. And it's an illusion. But that illusion is revealing to us that your visual system is computing motion using this type of correlation. So we could use the same illusion on the fly. And so if you do that, you find that everything is reversed on the fly. So basically, this really demonstrates that fly respond to odor motion illusion using this correlation-based type of algorithm. Okay. Now, can the olfactory system periphery detect this signal that would enable this motion? So in order to detect this motion, you have to be able to detect tiny differences in the signal between the two antennae. No? And so in the olfactory world, in the visual world, there's a classic type of model that have been used to describe and study these type of responses, which is called the Hassenstein-Reichardt two-point correlator. Basic idea is that you have two sensors that are spatially separated, and downstream for one sensor, you have a delay, and downstream for the other sensor, you don't have delay, and then the two signals are multiplied, or they interact non-linearly, basically. And so if you have the signal coming from left to right here, the signal will hit this one, but it's delay, and then when it hits this one, the two coincide, and you have a signal. But if it was moving from right to left, it would go down here and hit this one after. And then because there's the delay, you can have multiplication by 0 and you get no signal. So this will detect right moving motion, no? But not left. And so the fly has 2 antennae. And so we hypothesize that basically there should be some sort of Reichardt correlator inside the brain of the fly that perform this type of computation in a symmetric way. And so then we know the turning bias of the fly that we measure, and we know the orientation of the fly and the velocity of the bar. So we can relate the time for the bar to cross the antenna to the turning bias. And with that, we can extract a characteristic timescale of this delay that you would need for this type of computation. And it's on the order of 20 milliseconds, which is very close actually from what has been measured in the visual system. So that's encouraging. The next question is the olfactory periphery, basically those sensors here, are they able to encode the signal quickly enough and precisely enough such that you can maintain this information? And for that, we can measure the response. So this is the fly, this is the antenna, it's covered with little hair. And we can, in each one of those hair, you have 2 neurons, and the dendrites of the neuron, you have these olfactory receptors. And we can plant an electrode at the base here and then measure the response of those neurons. And so you get those spikes here as a function of time. And you see that they're so these are different ORN, different neurons, and we're presenting exactly the same signal multiple times. And you see that it's very precise. There's about 5 millisecond jitter. No? And so if you then use this type of data and look, and you take one of those measurements and another one, and then you shift them in time to introduce delays, you can calculate what would be the output of the HRC, and you find that basically you can detect motion left and right as long as the delay between the 2 antenna is on the order of like 20 milliseconds. Not when you hit 10 milliseconds is too fast, but the system can detect those signals. I'll pass over that. So then how do the fly combine wind direction and odor direction? So we know that they can detect the wind from previous studies, and now we discovered that they can detect also the motion of the odor. So you can do an experiment where you have wind and you flash the whole arena with a signal that illuminates the 2 antennas simultaneously. So there's no delay between the 2, okay? So this fly just knows there's odor, but doesn't know where it comes from. They cannot detect the odor motion. And so this is, if you want, the response to the wind, given that there's an odor, as a function of the orientation of the fly, and you get this kind of cosine type of response, reorientation. You can do, without wind, the response to only the odor, and you get a similar cosine. So the zero hypothesis would be that the fly, sum those 2 linearly. So we thought maybe they're just going to add those 2 into this. So if it linearly sum and take basically the sum of those 2 vectors as a new direction to go. And turns out that the data is very, very much matching this prediction. So then we can change the orientation of the wind and the motion of the odor. And in this case, you see that the sum is in between, and you see that the data is also in between the 2. And finally, we can make one against the other, and they kind of cancel each other, not perfectly, but basically you see that the bottom line is that a linear sum of those 2 signals is a very good zero-order model for how the flies are integrating these 2 signals. And this is become important to think about now. How is odor motion used to navigate? So when you navigate this type of signal, you have to think about the structure of the signal. So about 100 years ago, J. Taylor showed that the dispersion of a tracer in turbulent wind is governed by this continuous motion of the turbulent motion of the air, no? And so you have this correlation time of that random walk of those packets of odors. And so you can make a simple, instead of making a massive fluid mechanics type of simulation like I used to do in astrophysics, a simple model of this, you can generate packets of odor at a Poisson rate, and then have them do this correlated random walk, and at the same time, those packets diffuse due to molecular diffusion, so they become a bit larger. No? So there's molecular diffusion dm, but there's turbulent diffusion due to this correlated random walk. And this turbulent diffusivity is much larger than the molecular diffusion, which leads to this dispersion perpendicular to the main motion of the packet. No? And then if you look in this type of simulation, the mean wind direction is still always pointing from left to right, but the mean odor velocity field is pointing at an angle away from the center line. And so this suggests that basically this odor velocity field is providing extra information that's not present in the velocity field. That provide information about where is the center line of the plume. And so if you navigate, you don't have to just now turn upwind, but you could turn upwind and towards the center line of the plume, which should help a lot when you navigate. And you can also go a little bit further in this analysis. You could say, okay, let's now assume that I have a 2-point correlator detector, no? And so I define like a correlation function using this phi function, which is the concentration at a time, at a position x, y plus delta y, and a time t plus delta t, times the same with the minus and minus, and then minus the mixed case. And then from there, you can calculate what would be the correlation signal that you get. And you see that there's a dependency on the velocity of the packet, yi dot. No? And so from this information, from this signal, you can extract the velocity of the packet. And also, one has to remember that you have the packet spreading that is also important. So if this dp is too large, you can wash out the signal. But for this kind of simulation, we see clearly that you can have a positive sign here and a negative sign. So clearly, if you have this just 2-point correlator, you can tell if you're on the right or the left of the antenna. So now we can test if this is true by doing this experiment. So now we're going to put wind from left to right and bars that are moving away from the center. And so on the left, the fly should be able to navigate better than the right, because the right, the motion is opposed to what you would expect. No? So if they move against this motion, they're basically going to moving away. And if you look at how the fly navigate, you get that. You see how here the flies are basically following this edge and end up here. Well, like here, when they reach the end here, they kind of get lost. And actually, if you look at the number of flies that are in the front here, there's a clear difference between this case and this case. And more interestingly, you see that the distribution of the flies near the center, the front of the plume here, is much more peaked in this case than in this case. So there's clearly this motion reverse direction is confusing to the fly. But we can actually do better than that. What we can do is that we can take this smoke plume that we recorded and we can project them, I was saying before. But we're going to project them in 2 ways. On the left, we project them in normal. On the right, we're going to inverse time. And so you reverse the correlation when you do that, but you keep the rest of the statistics the same. And so in this case, the fly should do worse than in this case. And if you look at the performance, so the gray line are the flies that didn't make it, and the white one are the flies that made it. And the statistics are clearly, the success rate of the real flies to this is is massively different for just this tiny difference. Gradient and so on are the same, intensities are the same. So this is what I wanted to tell you, is that we discovered that there's a new sense here that flies seems to be able to do, which is detect the motion of odor with the olfactory system. And because it's such a simple, you just need two sensors and extract correlation, we think it's very likely that many animals actually are able to do that. The odor motion detection obeys a correlation-based algorithm that is equivalent to the one in the visual motion detection, which is also very interesting because those are different brain regions, but somehow the two are able to basically perform the same computation. If they do it the same way, we don't know, but this is going to be something interesting to look at. No? Also, odor motion provides a local directional cue that's complementary to the wind direction. And that's very interesting, not just for animals, but also if you build robots. So if you're building autonomous navigating robots, you do want to add the odor motion sensing capability to your robots. And actually in the paper, we added simulation of robots, very simple robots, turn right, turn left, etc., based on the sum of the direction of motion, and they do much better. You know? And it's very clear that the flies are actually exploiting this odor motion during olfactory navigation. So if you want to learn more about this, you can read those type of papers. And this, again, was done by Nirag, Mahmoud, and Damon Clark is my colleague who's an expert in visual motion detection, which shared with us all of those awesome different illusion patterns that we use in this study. Okay, thank you.